All right, so today we're going deep on ribosomes.
Ribosomes.
Yeah, those tiny little machines that build all the proteins in every living thing.
Right.
And we're doing this one thanks to you guys.
Oh, excellent.
Our listeners suggested it, and we've got excerpts from Gene Machine, which is Venki Ramakrishnan's own account of the race, to figure out these incredible structures.
Oh, that's cool. I've heard that's a great book.
It is, and it's not just like a dry science story.
Oh, right.
They're talking like rivalries and near disasters.
Oh, wow.
And even a few polar bears along the way.
Polar bears. I'm intrigued, but first, can you remind me why figuring out the ribosome was such a big deal?
Yeah.
I vaguely remember them from biology class,
Sure.
but it's all a bit hazy now.
Oh, trust me, this is way more exciting than high school bio.
Okay.
So see, before scientists could actually visualize these things,
Yeah.
understanding the ribosome was a real puzzle.
Okay.
And some really big names in science, even Francis Crick,
Oh, wow.
you know, the DNA guy, thought it was going to be easy peasy.
He actually called it a trivial problem,
Yeah.
which in hindsight is pretty funny,
I know, right?
considering it took decades of research and a Nobel Prize to finally crack it.
Yeah, trivial, it was not.
No.
So what made it such a tough nut to crack?
Mhm.
Was it just that ribosomes are super complex?
That's part of it, yeah.
Okay.
Imagine like a tangled mess of proteins and RNA molecules
Yeah.
all jammed together in this
Yeah.
intricate 3D structure.
Right.
But there's also this kind of chicken or egg paradox at play.
Yeah.
Ribosomes make proteins,
Right.
but they're also made of proteins.
Okay.
So how did the first ribosome even come about?
Right.
It's a real head scratcher.
Yeah, that classic dilemma.
Yeah.
Makes sense why scientists were initially stumped.
Yeah.
But the source material mentions this debate about whether it was the RNA or the proteins that were really running the show.
Yeah.
What's the story there?
So for a long time, scientists really focused on the proteins,
Okay.
thinking they were the key players. But there were a few, like Harry Noller,
Yeah.
who became this strong advocate for RNA's importance.
Okay.
Ramakrishnan actually calls him the Sage of Santa Cruz.
Sage of Santa Cruz.
Yeah,
That definitely sounds like someone worth knowing.
it does.
So was he like a lone wolf going against the grain?
Well, he definitely had a strong personality,
Okay.
but he was also a brilliant scientist.
Right.
He did some early experiments that really hinted that RNA might be more than just this passive scaffold.
Okay.
But to really understand what was going on, they needed to figure out the ribosome structure,
Right.
which meant turning to X-ray crystallography.
X-ray crystallography, right?
Yeah.
It rings a bell, but can you refresh my memory on how that works?
Sure.
It's something about crystals and X-rays, right?
It is.
Okay.
So imagine you're trying to figure out the shape of a statue in a dark room.
Okay.
You could shine a flashlight on it,
Yeah.
and by looking at the shadows that it casts,
Right.
you could start to get an idea of its form. X-ray crystallography works in a similar way.
Okay.
So you take your molecule, in this case the ribosome,
Right.
and you try to get it to form a crystal,
Yeah.
like a perfectly ordered 3D arrangement.
Okay.
Then you shine X-rays on the crystal
Right.
and look at how the rays diffract or scatter.
Okay, so the diffraction pattern is like the shadow of the molecule.
Exactly.
And that tells you the shape.
Exactly. It's like a fingerprint that reveals the arrangement of atoms within the molecule.
That's pretty wild.
It is.
But crystallizing something as complex as a ribosome,
Yeah.
that just seems like mission impossible.
It was an enormous challenge.
Yeah.
Think about it. Tons of these tiny molecules,
Right.
all needing to line up perfectly in 3 dimensions.
Wow.
And at this point, scientists weren't even sure if all ribosomes from a particular organism were identical.
Oh, wow.
It took pioneers like Ada Yonath and Heinz Günther Whitman
Okay.
years of painstaking work to even get tiny, imperfect crystals.
So the early days were filled with frustration. So were there any breakthroughs, or was it just like endless trial and error?
There were some intriguing discoveries along the way.
Okay.
For example, they found that ribosomes from extremophiles,
Okay.
those organisms that live in really extreme environments,
Okay.
like hot springs or the Dead Sea,
Oh, wow.
they actually form better crystals.
Really?
It turns out that these ribosomes are just more robust,
Okay.
which helps when you're blasting them with X-rays.
Survival of the fittest,
Right.
even at the molecular level.
Exactly.
Makes sense, but wait.
Yeah.
Didn't the source material mention something about a researcher whose work turned out to be well less than truthful?
You're talking about Haskell parodies.
Haskell parodies.
Yeah, he was like a rock star in the crystallography world,
Okay.
claiming to have crystallized all sorts of important molecules, including ribosomes.
Wow.
And then people started noticing inconsistencies.
Oh,
And it turned out that a lot of his data was fabricated.
wow. Talk about a setback.
It was huge.
Not just for the science, but for the trust within the research community.
Exactly.
I can only imagine how demoralizing that must have been.
Yeah, it was a huge blow.
Yeah.
People became much more cautious,
Right.
and it just made the race to solve the ribosome even more challenging.
Right. So where does Venki Ramakrishnan, our author,
Yeah.
fit into all of this? Was he involved in this early crystallography work?
Actually, his path to ribosome research was quite unexpected.
Oh, okay.
He started out as a physicist,
Oh, wow.
but stumbled upon a Scientific American article about ribosome research
Interesting.
that completely captivated him.
Okay.
That's how he ended up switching fields entirely
Wow.
and doing a postdoc with Peter Moore at Yale,
Okay.
using a technique called neutron scattering to study the ribosome.
Hold on. He switched from physics to biology just like that?
It was a big risk.
That's a pretty bold move.
It was, but he was clearly drawn to the challenge,
Yeah.
the mystery of the ribosome.
Right.
You have to admire that kind of scientific curiosity,
Yeah.
the willingness to just jump into the unknown.
I'm sensing a theme here.
Yeah,
Taking risks, pushing boundaries.
absolutely.
But what about this neutron scattering approach?
Yeah.
Did it pan out?
It was promising at first,
Okay.
but eventually it just hit a wall. It just couldn't provide the level of detail they needed to crack the ribosome structure.
Right.
And that's when Ramakrishnan's journey takes another really interesting turn.
Okay.
Frustrated with the lack of progress,
Yeah.
he decides to spend a sabbatical at the LMB,
The LMB.
yeah, the mecca of crystallography,
Oh, wow.
to learn X-ray crystallography himself.
Wow. So he basically decided to learn a whole new, incredibly complex technique on the fly.
It was a long shot.
That's dedication.
It was. But he was determined to be at the forefront of ribosome research.
Right.
And he knew that X-ray crystallography was the key.
Talk about a pivotal moment. It
Yeah.
really shows how a single decision can change the course of a scientific career.
Absolutely.
And in this case, maybe even the course of scientific history.
It might have, yeah.
But we're just scratching the surface here.
I know.
We've got crystallization challenges, intense competition, and those elusive high-resolution structures still ahead of us.
And let's not forget those polar bears.
Right. The polar bears.
I'm still waiting to hear how they fit into all of this.
Okay, stay tuned because things are about to get really interesting.
They are. So Ramakrishnan gets to the LMB, eager to learn all this X-ray crystallography stuff.
He walks right onto the field just as the game is getting good.
I know, right?
It's exciting. The source material mentions Ada Yonath making a big breakthrough around this time. Something about 3 angstrom resolution crystals.
Yeah.
What's so special about that?
Well, that was a major milestone. 3 angstroms means you can start to see individual atoms within the molecule.
Oh, wow.
And Ada had managed to grow these incredibly detailed crystals of the large ribosomal subunit, the 50S, from salt-loving bacteria called Haloarcula merismortui.
Okay.
It really seemed like a
That's
full atomic structure of the ribosome was within reach. But there was still this huge obstacle, a problem that had always plagued X-ray crystallography: radiation damage.
Right. I remember reading about that you shine intense X-rays at these fragile crystals, and they basically start to disintegrate before you can collect all the data you need.
Exactly.
It's like trying to take a picture of a snowflake with a blowtorch.
That's a great analogy.
It really is.
But just when things were looking bleak, a truly game-changing solution emerged: cryocrystallography.
Cryocrystallography.
Yeah, the idea was to flash freeze the crystals in liquid nitrogen,
Oh,
essentially hitting the pause button on the radiation damage.
that's brilliant. Sometimes the simplest solutions are the most elegant.
Yeah.
It's amazing how a technique like that can just revolutionize an entire field.
It really was a turning point.
Yeah.
Cryocrystallography opened the door for high-resolution structures of all sorts of complex molecules.
Right.
But back to Ramakrishnan, he's at the LMB absorbing all this crystallography knowledge.
Right.
And he makes a decision that I find quite interesting. He decides to focus on the smaller ribosomal subunit, the 30S.
Why the shift in focus? Was it just that the 30S was smaller and maybe easier to work with?
It was more strategic than that. Ada Yonath and several other groups were all focused on the larger 50S subunit.
Okay.
So Ramakrishnan saw an opportunity.
Uh-huh.
He felt like the 30S was a more manageable target and that solving its structure would still be a significant contribution to the field.
So he saw a path less traveled and decided to take a chance. Sometimes in science it pays to be a bit of a maverick.
Right.
But wasn't there a risk that if he solved the 30S structure, it might be seen as less important than the 50S?
That's a fair point.
Yep.
But remember, the 30S subunit is still incredibly important.
Yeah.
It's the part of the ribosome that binds to messenger RNA
Okay.
and makes sure that the genetic code is read correctly.
Right.
Without it, protein synthesis would be a complete mess.
Right. Okay. That makes sense. So Ramakrishnan sets his steets on the 30S. But how do you even begin to solve a structure this complex?
Mm.
It's like a giant 3D jigsaw puzzle with millions of pieces and no picture on the box.
It is a puzzle, and a very challenging one at that. One of the biggest hurdles in X-ray crystallography is something called the phase problem.
The phase problem.
Yeah, basically to calculate the structure from the diffraction pattern,
Yeah.
you need to know not just the intensity of the diffracted X-rays, but also their phase, which is a measure of their wave-like properties.
Okay.
But unfortunately, the detector can only record the intensity, not the phase.
So you're missing this crucial piece of information. It's like trying to solve a Sudoku puzzle where half the numbers are missing.
Yeah.
How do you even begin to approach that?
So they used a clever trick called isomorphous replacement.
Isomorphous replacement.
Yeah, the idea is to soak the crystals in solutions containing heavy atoms like mercury or gold.
Okay.
And if these heavy atoms bind to the ribosome in specific locations, they'll change the diffraction pattern in a predictable way.
Okay.
And by comparing the diffraction patterns with and without the heavy atoms, you can start to figure out those missing phases.
So the heavy atoms act like landmarks, helping to pinpoint the positions of other atoms in the structure.
Exactly.
But the source material mentioned heavy atom clusters.
Yes.
What's the difference?
That's an important detail.
Yeah.
So Ramakrishnan realized that instead of using individual heavy atoms, which can be really tricky to work with,
Okay.
he could use clusters of heavy atoms.
Okay.
It was a risky strategy, but it turned out to be crucial to their success.
It's like using a bigger boulder marker to make those landmarks stand out.
Right.
I can see how that would be helpful. But I'm sensing some competition here. Wasn't there another group at Yale also working on the 30S structure?
Absolutely. This is where the rivalry between Ramakrishnan's group and the team at Yale, led by Peter Moore and Tom Stites, really comes to the forefront.
Okay.
Both groups were using similar approaches,
Uh-huh.
and they were just neck and neck in the race to get those high-resolution structures.
Okay, so the pressure is on. This is getting intense.
Yeah.
Were there any, cloak and dagger moments in this rivalry? Were they spying on each other, trying to sabotage experiments?
Not quite that dramatic.
Okay.
But there was definitely a sense of urgency.
Great.
Both groups knew that the stakes were high,
Yeah.
and they were constantly leapfrogging each other.
Oh, wow.
Each new development pushed the other team to work harder and faster.
I bet the atmosphere in both labs was electric. Everyone pushing themselves to the limit, the excitement building. So they're collecting data, refining their crystals, the tension is mounting. What happens next?
Well, this is where things take a rather unexpected turn. Remember those polar bears I mentioned earlier?
Oh, right. The polar bears have been waiting for this. How do they fit into the story?
It turns out that ribosomes from Arctic hibernation organisms were particularly well-suited to X-ray crystallography.
What?
Their ribosomes were more stable, able to withstand the intense X-ray beams for longer periods.
Wait, so they were using ribosomes from polar bears?
It's a reminder that scientific breakthroughs can come from the most unexpected places.
That's incredible. Who would have thought that these majestic creatures would hold the key to understanding such a fundamental biological process?
I know, right?
This is what I love about science. It's full of surprises.
It is.
But where do those suitcase and bombs come in? The source material mentions something about that.
Ah, yes. That's one of my favorite anecdotes from the book. They had finally secured some precious time at the Advanced Photon Source, or APS, at Argonne National Laboratory,
Okay.
which had one of the most powerful X-ray beams in the world.
Wow.
But getting their precious crystals there in perfect condition was a logistical nightmare.
Yeah.
They had these massive dewars filled with liquid nitrogen to keep the crystals frozen.
Okay.
And one was this huge rectangular container.
Hence the suitcase.
Exactly. And the other two dewars were these cylindrical containers with rounded tops, which, to their amusement, looked a lot like bombs.
I can only imagine the looks they got from the TSA agents.
Right.
Oh, these.
Just a suitcase and a couple of bombs.
Yeah.
Nothing to see here. But seriously, they get to Argonne. They've got limited time with this super-powered X-ray beam. Did they manage to crack the structure? That's what we'll find out in the final part of our deep dive. The anticipation is killing me.
Okay, we're back, and the suspense is killing me. Our Ribosome hunters have arrived at Argonne National Laboratory with their suitcase and bombs, ready to unleash the power of the advanced photon source on their precious crystals. This is the moment of truth. So what happened? Did they finally solve the structure?
They were working around the clock, analyzing data, tweaking their crystals, pushing their equipment to the limit.
Right.
Ramakrishnan describes this almost military-like precision in their approach,
Wow.
working in 12-hour shifts, barely sleeping, driven by this intense focus and determination.
Wow.
And then it happened. They started to see these clear, beautiful maps of the 30S subunit emerging from the data. They could trace the RNA helices, the protein backbones.
It was all coming into focus.
It was.
I can only imagine that feeling, years of work, all those setbacks and challenges culminating in this moment of revelation.
It must have been exhilarating.
Yeah.
It's one of those moments that every scientist dreams of. That sudden flash of insight when all the pieces of the puzzle fall into place.
Right.
And it's a testament to their hard work, their ingenuity, and their absolute refusal to give up.
But having those maps was just the beginning, right?
Right.
They still had to build the actual atomic model from this intricate 3D electron density map.
Yeah.
That seems like a whole other level of complexity.
You're absolutely right. Building the model involved meticulously fitting the known protein and RNA sequences into the electron density maps, making sure every atom was in the right place.
Okay.
And it was during this process that they made a truly groundbreaking discovery.
What's that?
They realized that the RNA, which for so long had been considered just a structural scaffold, was actually the catalytic core of the ribosome,
Really?
the part that actually carries out the protein synthesis reaction.
Wait, so you're telling me that the RNA, this molecule that everyone thought was just a supporting player,
Yeah.
turned out to be the star of the show?
Precisely. It was a huge discovery, and it completely changed how scientists thought about the role of RNA and the origins and evolution of life.
Wow.
It lent strong support to the RNA world hypothesis,
Right.
which proposes that early life was based on RNA, not DNA.
That's mind-blowing.
It
It's
is.
like the underdog suddenly winning the championship.
Yeah.
But they weren't out of the woods yet, were they? They still had the Yale group breathing down their necks,
Right.
and Adayonath working on the 50S subunit.
The competition was fierce, and Ramakrishnan knew they couldn't afford to let up. They had to get the results published as quickly as possible.
Oh, wait.
He describes this frantic rush to write the paper and submit it to Nature, all while keeping one eye on the competition.
Yeah.
And then they made a rather bold decision.
Yeah.
They decided to present their findings at a conference before the paper was even published.
Hang on, why would they do that? Weren't they worried about someone stealing their ideas?
It was a calculated risk. By going public, they were staking their claim, establishing their priority in the right. It was like firing a shot across the bow of their rivals.
It's a bold move, and it sounds like it paid off. So what was the reaction?
It created a huge buzz within the scientific community.
Yeah.
Everyone knew the ribosome structure was imminent, and the race was on.
Wow.
The pressure intensified, and soon after, both Ramakrishnan's group and the Yale group published their 30S structures in Nature just weeks apart.
Oh, wow.
Adayonath's 50S structure followed soon after.
So a three-way tie for the win.
In a sense, yes. It was a collective victory, a demonstration of the power of the scientific method and the relentless pursuit of knowledge.
Wow.
But the story doesn't end there. Solving the structures of the individual subunits was just the beginning.
Okay.
The ultimate goal was to understand how the two subunits came together to form the complete ribosome, the 70S,
Right.
and how it interacted with all the other molecules involved in protein synthesis.
It's like they had assembled the pieces of the puzzle, but
Yeah.
now they had to figure out how they all fit together and how the whole machine actually worked.
Exactly.
Hmm.
And in the years that followed, Ramakrishnan, along with many other scientists, continued to refine the structures, capture the ribosome in different states of action, and really piece together the intricate molecular dance of protein synthesis.
Wow.
It's a story that's still being written today.
It's incredible to think that we've gone from trivial problem to these detailed atomic-level snapshots of one of the most fundamental processes in biology.
No, it's amazing.
And you mentioned something about a magnesium blender.
Oh, yeah.
What happened there?
Oh, that's a classic example of how even the best scientists can make mistakes.
Sure.
While working on the 70th structure, Ramakrishnan's team accidentally omitted a crucial magnesium ion from their model,
Oh, no.
and it threw off all their calculations.
Yeah.
It cost them valuable time and could have set them back significantly in the race.
Wow.
Luckily, they caught the error and were able to correct it.
That must have been a heart-stopping moment. You're racing against the clock and suddenly you realize you might have made a critical error. Talk about pressure. But ultimately, it all worked out. And Ramakrishnan went on to share the Nobel Prize for his work on the ribosome.
Yeah.
And his memoir gives us this fascinating glimpse behind the scenes of scientific recognition.
Yeah, he's very open about the complexities of the Nobel Prize selection process and the inherent biases within the system.
Okay.
He acknowledges that countless other scientists whose contributions were crucial to the success of the ribosome project.
It's a good reminder that science is rarely a solo endeavor.
Right.
It's a collaborative effort built on the work of countless individuals, all driven by the same desire to understand the world around us.
And Ramakrishnan's story is a powerful testament to that spirit of collaboration, curiosity, and persistence.
Absolutely.
It's a story that reminds us that even the most challenging scientific problems can be solved with ingenuity, determination, and a bit of luck.
And perhaps a few polar bears along the way. This has been an incredible deep dive full of surprises, insights, and even a few laughs. I'm so glad we had the chance to explore this fascinating world together.
Me too. It's been a privilege to share this journey of discovery with you.
And to our listener, thanks for joining us on this deep dive into the world of the ribosome. Until next time, keep exploring, keep asking questions, and never stop learning.